Good morning to one and all. Now we have to proceed further. The next detector, ECD, or say electron capture detector, that we have to see in detail. Just I am sharing the screen. Does it visible or not? Please tell me. Yeah, is it visible?
Yes, sir.
Yes, sir.
Okay. See, electron capture detector, the diagram is shown in your that particular notes. ये डायग्राम आपके इसमें दिए है कि एक शेल्ड बॉक्स है, उसके अंदर यू कैन सी द रेडिएक्टिव सोर्स इज देयर. ओके, उसके बाद में हम इनलेट से सैंपल अंदर डालते हैं, फिर जो कुछ भी रेडिएशन या कुछ वहाँ पे जो इलेक्ट्रॉन क्रिएट होता है और से आयोनाइजेशन होता है, वो एग्जॉस्ट यहाँ पे दिया है, इलेक्ट्रिकल सर्किट यहाँ पे मेंटेन किया है. ओके, this all about given in your notes. Okay, that just you see the video, you will clear more idea.
Write a note on electron capture detector. The electron capture detector, or ECD, is based on the fact that the current strength decreases when compounds in the effluent flow capture electrons. In the ECD, the effluent gas is exposed to alpha or beta rays from the radioactive source. When the compounds in the effluent capture the free electrons, there will be a decrease in current, and this is measured in contrast to the measurement of increase in current in other ionization detectors. The ECD is a modification of ionization chamber used for detection of radiations. The effluent from the chromatographic column is exposed to slow electrons generated by the ionization of the carrier gas, which is either argon or nitrogen, by a constant flux of beta rays from the radioisotope. When an electron capturing molecule in the effluent gas captures an electron, the result is the formation of a negative ion of a much higher mass and a consequent reduction of current flow. The ECD is a very sensitive detector for certain compounds, such as halogenated organic compounds, but is sensitive to compounds like hydrocarbons. The detector is very useful in the detection of ultra trace amount of metals. It is also used for the analysis of insecticides and pesticides. So if you like this video, please click on the like button and also subscribe to my channel.
Okay, I think so you got this concept. See what happens generally in the ECD, the current whatever is there, that decreases when the compound present in the effluent, okay, um, capture the electrons. Now the electron source that is given to you, the beta rays or alpha rays are used as that particular electron source. In the bracket, the nickel foil, iron, titanium, then titanium, tritium. Okay, these three elements are given, which are used as a radioactive source. Okay, the compound in the effluent which captures the electron, and after capturing the electron, there will be the decrease in the current, and this will be measured in contrast to the measurement of increase in color in other ionization detection. Okay, so in this way loss in current is measured. What they are supposed to, loss in current is measured, then decrease in current is recorded as a negative peak on the recorder. These are the advantages and disadvantages. Same thing was there in that particular video. It is a very simple and that what we can say, that detector. Okay, it does not respond to the hydrocarbons. It is a drawback of this particular ECD. Okay,
Right.
here generally once again I will show in detail. See that particular flame ionization detector.
First of all, We would like to talk about how gas chromatography works before going into the details of the flame ionization detector. In gas chromatography, a gaseous mobile phase transports gaseous solids through a column as the one shown in the picture. As we can see in the diagram, the sample is injected before entering the oven and column. In order to move the sample through the column, we need an inert gas that serves us as a carrier. After leaving the column, it is that the solids flow through the flame ionization detector. In order for this process to work, the capillary column must be at an adequate temperature to produce enough vapor pressure for the solids to be eluted reasonably. Then, the detector will be at a higher temperature so we can make sure that all the solids are in the gaseous state. For gas chromatography, the size of the sample must be very small since the column just can handle very small volumes. The sample can be introduced into the process by using a syringe or a gas sampling valve. Now, let's talk more in detail about the functions of a flame ionization detector. A flame ionization detector is used to detect the ions produced by a flame. In order to be able to detect these ions, we need two electrodes, which are used, and the potential difference is applied. This diagram shows the different steps through which the eluent gas coming out of the column goes through. As shown by the pink section of the diagram, the eluent from the column enters the detector. At this time, hydrogen is mixed with the eluent so that it can go to a small jet where it is burned. This reaction shows what happens. Carbon atoms produce CH radicals which produce CHO plus ions in the flame. At the bottom of the flame, we have the negative electrode. In the area where the collection occurs, we have the positive electrode. High voltage is applied between the electrodes so that the ions produced in the flame can be collected. As shown in the diagram, in order for the detector to work properly, we need three different gas inlets. Through these three different gas inlets, the gases that are normally used are hydrogen for the combustion process, nitrogen for the carrier gas, hydrogen for the combustion process, nitrogen for the carrier gas, and air or oxygen to act as the combustion agents. Now, let's switch gears a little and let's talk about the advantages and disadvantages of using a flame ionization detector. There are many different advantages when using a flame ionization detector. First of all, flame ionization detectors are very sensitive to a wide range of components. Not only that, but it also works over a wide range of concentrations. A really good example of a flame ionization detector is the Autofilm Flame Ionization Detector, manufactured by AFC International Inc. This specific detector has a range of from 0 to 10,000 parts per million, and the detection limit is of 0.1 parts per million, which is pretty low, which is a really good detection limit for a gas chromatographer detector. By far, the flame ionization detector is the most useful gas chromatography detector. It is also the most commonly used in gas chromatography analysis. Now, let's compare two different detectors, the flame ionization detector and the thermal conductivity detector. Which of these two detectors is better, the flame ionization detector or the thermal conductivity detector? And yes, the answer is the flame ionization detector. And this is because the flame ionization detector has a much lower detection limit than the thermal conductivity detector. Now, let me clarify something. By better, we mean that it has a much lower detection limit for hydrocarbons than the detector that we have described previously in this presentation. Now, let's talk about the disadvantages. One of the disadvantages is that the sample use is destroyed since it is burned by the flame. However, it does not matter much that it's destructive since the detector requires only minimal volumes of sample. Another disadvantage is that the flame ionization detector is non-selective, so there is a potential for other compounds to interfere with this analysis. One last thing is that this detector responds to most hydrocarbons, but it's insensitive to non-hydrocarbons such as hydrogen gas, helium, nitrogen gas, oxygen, carbon monoxide, carbon dioxide gas, helium, nitrogen gas, oxygen, carbon monoxide, carbon dioxide, water, ammonia, nitric oxide, and hydrogen sulfide. Although these disadvantages exist, the flame ionization detector is still one of the best detection methods in gas chromatography.
Okay, then we have to talk about the qualitative and the quantitative analysis. These two particular things can be done in case of the GLC or the gas chromatography. See, in gas chromatography, you have to remember that it is widely used technique to check the purity of the organic compound, also to check the contaminants. Okay. Qualitative analysis of the sample involved determination of the total number of components present in the sample. You can see it will can identify number of the peaks in the chromatogram. Then the identification of the each component present in the sample. This is done by the retention time. अगर आपको retention time पता है तो आप क्या identify कर सकते हैं कौन सा component present है और जितने peaks आएंगे उतना क्या रहेगा उतने components present रहेंगे. Then characterization of each component of the sample. Okay, यहाँ पे भी दिया है same thing is given here. Then quantitative analysis, it involves the determination of amount of concentration of each component in the sample. तो उसके लिए क्या important है that quantitative analysis is based upon the comparison of either height or the area of the analyte peak with that of the one or more standards. Okay, the area under the peak is used for the determination of concentration of or the amount. The area under the peak of standard and the sample component is prepared. The area under peak is proportional to the amount of the component responsible for the peak. A standard upon A sample is equal to W standard upon the W sample. This ratio is compared. The relative percentage of the component in the sample can be calculated based on the total areas of all the peaks obtained in the chromatogram. Therefore, percentage component is equal to AI upon summation AI into 100, where AI is the area of the single peak of the chromatogram. This quantitative analysis you can done how much amount is present. Okay, qualitatively means which component, which species is present. Okay, these two things you can do with this particular gas chromatography. Applications, there are the number of applications of this gas chromatography. Chromatography is applicable only to those components that can be vaporized at the temperature of the operation. Any number of components in the sample can be identified and determined by this method. It is very useful in the separation of reacting mixture as well as complex natural product. This technique can be successfully used for the separation of chemically identical compound. Example, isomers of the compounds you can identify, you can that differentiate. Okay, low boiling gases like nitrogen, hydrogen can be separated with the large column. Analysis of essential oils can be carried out. Analysis of this essential oil also carried out. ppb level concentration, see parts per billion of some toxic compounds. You know, जो लोग ड्रग्स लेते हैं तो उसका क्रोमेटोग्राफी भी करते हैं. ओके, आज में उसका सैंपल का क्रोमेटोग्राफी किया जाता है कि ट्रेस अमाउंट में भी उसने अगर ड्रग्स लिया होगा तो यू कैन इजीली आइडेंटिफाई. या क्लोरोफॉर्म तो बताया है आपको Amino acids, proteins, peptide, lipids, these types of the biomolecule can be separated and determined. Terpenes, steroids, alkaloids from the natural product sample can be separated and estimated. Materials of the plant origin can also be separated. Different herbal extract is used as a part of the herbal drug formulation can be analyzed by this method. Automobile exhaust gases, a main pollution hazard, can be analyzed by this method. It is also used in the separation of the radioactive products, and it is also used to study the reaction mechanism. ये भी उसका application है. There is a lot of applications. आपको जैसे ध्यान में रहेगा that you can write. Okay, this is all about this particular your gas chromatography. Okay, anybody is having any problem? Anybody is not understood any concept regarding gas chromatography, you can ask now. Then in the next lecture, we have to talk about the ion exchange chromatography. Yes, anybody is not understood the principle or anything of this particular gas chromatography, please tell me. I will repeat that part again. Umar, have you
Yes,
got?
sir. Yes, sir.
Tanzil, Taski, now anybody is having any problem, you can ask.